Chapter 12 is our first chapter in the nervous system, and in reality, we are going to be in the nervous system until the end of our semester. There is a lot to learn in this very complicated system. So this chapter is really going to serve as an overview. We're going to take a broad look at the layout of the nervous system. We'll look a little bit more in depth at some of the cells that are involved in the nervous system, and then particularly we'll spend some time on some neurophysiology, looking at how neurons actually pass messages to cells to tell them what to do. So starting out with just a very brief overview here, what I want you to notice in this first picture in the chapter is that we separate the nervous system into the central nervous system, which includes the brain and the spinal cord, and then the peripheral nervous system, which is basically everything else. What I want you to realize is this is really an arbitrary distinction. A lot of times what we see are humans naming things, labeling them, trying to organize them to help us better maybe comprehend, memorize, understand the pieces parts. But I do want you to realize that these are fully integrated, they're working as one system. Same thing on this particular chart, we'll notice that we separate the nervous system into two main branches, a sensory or afferent division that's actually capturing information from throughout the body. This would include things like our special senses, sight, sound, balance, etc. In fact, we have an entire chapter on the special senses, right? But all of that sensory information is going to travel via neurons up this afferent division to the central nervous system, who really gets to decide what to do with that information. So we have information from the special sense organs. We have a lot of receptors in the viscera, in the guts, right? So receptors that tell us when the stomach has stretched, that's how we know we're full, etc. And then also somatic sensory receptors. These might be receptors on the surface of the skin that we talked about in the integumentary chapter. We'll get into some more detail here in the nervous system. And then we also briefly mentioned in the muscular system or in the chapter on articulations that we actually also have a lot of receptors, whether they're embedded in the skeletal muscle or in the tendons or in the joint capsule, to really provide, again, the brain or spinal cord information about the position of the joints, the stretch of the muscle, those sorts of things, right? This afferent division is always sensory information going towards the CNS. When we talk about the central nervous system, we're really looking at information processing, integrating all this information, deciding what it means, and coming up with a plan of what the body should do, right? And so the output from the central nervous system is our motor commands, or what we call the efferent division. You'll see these terms again in AMP2. Afferent, right, is going towards the central nervous system. Efferent, maybe exiting is a better word than away, exiting the central nervous system, right? And this is the branch of the nervous system that's going to be telling the body how to respond. So we see the effectors down here. This is the response. Again, dividing the nervous system, trying to make it make more sense, we can divide that efferent division into the SNS, the somatic nervous system, and the ANS, the autonomic nervous system. Now, soma means body, which is maybe a nondescript term in human anatomy physiology, but what we're looking at here on the somatic nervous system, those messages are going to the body, they're going to the skeletal muscle, right? So think about, the brain has decided you should in fact raise your hand. You know the answer, raise your hand, right? And it sends that motor output to coordinate the skeletal muscles in a way that gets you to do its bidding. On the other half, the autonomic nervous system, this is going to be involuntary, right? We consider this voluntary. The brain made a decision and told your skeletal muscles what to do. The autonomic nervous system deals with a lot of other organs that are under unconscious or involuntary control. We further break the autonomic nervous system down into the sympathetic division and the parasympathetic division. You may have heard of this before. The sympathetic nervous system is referred to as your fight or flight response. The parasympathetic nervous system basically does the opposite. Instead of getting ready to fight, right, it calms everything down. We tend to think of this as encouraging rest and digest. Let me show you what I mean. Oh boy, I got things everywhere here. So I pulled this from a different textbook, but I thought it had some nice images for parasympathetic versus sympathetic nervous system. Notice in the fight or flight response, everything is all about getting ready to deal with a dangerous situation. The pupils are dilated, you're trying to take in more information, right? Heart rate, breathing rate are both increased. We're secreting epinephrine. We're saying now is not the time to pee. Now is not the time to try to digest food or reproduce, right? So if you had skeletal muscle on here, you'd see increased blood flow and things like that. Okay, so fight or flight. Rest and digest, notice it slows the heart, it slows the breathing. And this is what we're stimulating now, right? Is digestion, the urinary system, the reproductive system. So they're kind of opposite branches. But again, I would encourage you to think about this as an integrated system working all as one. This is really the big overview of the nervous system, and we'll delve more into a lot of these pieces, parts, as we move along. Okay, before we go further though, let's spend a little bit of time talking about the tissue that makes up the nervous system. So you may notice that I pulled this from chapter 4, our chapter on tissues, and we said nervous tissue is one of those 4 main types of tissue. It includes the neurons, which is what we'll spend most of our time talking about, but also the neuroglia, the support cells, the ones that take care of and insulate and protect the neurons. When we look at these neurons, your book gives you this kind of cartoonish image to start things off, and then we'll get a little more complicated, right? But they want you to notice the dendrites, right? These are short projections on one end of the cell body, and these dendrites are actually collecting information. They're the receiving end of the cell, right? So the dendrites, this is the receiving, I before E except after C. Okay, and so they're collecting all this incoming information. The dendrites are actually incapable of propagating action potentials. They do not send electricity down the cell. They're going to have what are called graded potentials. And these are more short-lived, localized messages that they'll send. Because really, they're trying to get the message here to the cell body. The cell body is sometimes also called the soma. And this is going to be the part of the cell then that is gathering all that information and kind of deciding what to do with it. If that message is loud enough from the dendrites, we can use this axon to carry a message in the form of electricity, in the form of an action potential, right? Axons do propagate action potentials, just like we saw in skeletal muscle, and they can send that message down then to the axon terminals. What they're trying to show you here with telodendria is kind of a reminder that you can have one long axon that then branches. The way you already know this is true is in the muscular system we talked about the motor units, where you had a single neuron and all the muscle cells that it innervated. That means that a single neuron, a single axon, has to be able to branch to go ahead and reach all of those cells. let's look at this in a little more detail. Okay, so they're showing us a couple things in this image, right? Again, we see all these dendrites. They've put a lot more detail now into that cell body, right? They're showing us a couple things here about the neurons and about their metabolic activity. So they're pointing out part of the reason the cell body stains gray. So if you've heard of gray matter on the brain, right, it's going to be a lot of cell bodies. The reason it kind of stains gray are these Nissl bodies, and this is a lot of rough endoplasmic reticulum, some free ribosomes as well, but that really tells us that we are producing a lot of proteins in this neuron, right? Those might be neurotransmitters that we need to use. Those might be new sodium channels, potassium channels, right, along the distance. So we're making lots of proteins. And that endoplasmic reticulum, we're also able to make lots of new lipids. We can repair the membrane and that sort of thing. Okay, now I mentioned before that the dendrites do not carry an action potential, right? They use graded potentials to communicate here with the cell body. Oh, sorry. The other thing they're pointing out are there's an awful lot of mitochondria in neurons as well. They are extremely metabolically active. I forget the exact number, but the brain takes an awful lot of your oxygen and the nutrients that you have to keep running. Right? If you feel like you're trying to learn all of this and your brain hurts and it drains you, absolutely, the brain is working hard, trying to make new connections, make new synapses to know things. Okay, a little more detail here on the axon. They're pointing out here that as we leave the cell body and we're narrowing into the axon, we reach a portion of the cell called the axon hillock, right where it's kind of tapering down. This is going to be the portion of the cell where we actually are able to trigger an action potential, right? Basically, the big difference is that only the axon hillock and down through the axon have voltage-gated channels. And the voltage-gated channel, if you recall, when that depolarization hits, it opens the next sodium channel, more sodium comes in, the depolarization moves down. Without the voltage-gated channels, you can only create graded potentials. So we see the voltage-gated channels and therefore our action potentials on the axon. Okay? Again, they're showing us the axon, the telodendria, those branches, and recall that the very end of each branch is that axon terminal, where we actually have a synapse, a space between that adjacent cell, right? And we're going to do the whole neurotransmitters have to cross that gap. Now, when we were in the muscle chapter, we had a neuron and we had a skeletal muscle, right? We called this the neuromuscular junction, right? A specific name for that synapse. Now in the nervous system, we have a lot of times where one neuron is talking to another neuron, and we have to have a way to distinguish which is which. And so you may notice that they're referring to this neuron as the presynaptic, right? Remember, our synapse is that little gap. So this is the presynaptic neuron, that is the postsynaptic neuron. Okay. Another thing to point out, they're showing you here action potentials only run in one direction. Absolutely 100% true all of the time. You also have substances that are moving through the cytoplasm of the cell. These might be things like mitochondria. They actually reproduce up here in it's asexual reproduction, but they reproduce up here in the cell body, but they'll travel down the cytoplasm. You actually need a lot of ATP at that axon terminal, so we'll have mitochondria actually getting carried down to those terminals. It's really crazy. They have some cartoons online that kind of show you they're traveling down the cytoskeleton, and you have these proteins in cells. They're called motor proteins, and they literally just walk along that cytoskeleton, right, some of the tubules, and they're just dragging a mitochondria behind them. Really fascinating, right? So that's substances that are getting sent down the axon through the cytoplasm, mitochondria. You make a lot of your neurotransmitters, right? That's all the ribosomes creating these neurotransmitters, but you need to transport them as well. They go down the axon. When we move the substances in that direction, it's called anterograde transport, and that's going the same direction as an action potential. We said no action potentials can go backwards, but you can get what's called retrograde transport in the cytoplasm. So as you get worn out organelles or broken down neurotransmitters that you want to recycle, retrograde transport can take them back up the axon and into the cell body where they can get processed. Now that becomes interesting because if you think about something like rabies, you can get bit on the arm, right? But if that virus makes it into the neuron, it can use that retrograde transport to actually get back into the cell body, which often is in the central nervous system. Okay. Now, as far as where you will find cell bodies versus axons, right? Cell bodies, remember, they tend to stain gray because of all of these ribosomes, and so we'll end up calling this gray matter. The axons tend to be insulated. We have specialized cells, some of our neuroglia that we can talk about, that actually put layers of lipid, right, insulation around those axons. This increases the speed of the transmission of an action potential. And so we talk about that, the portions of the nervous system that contain a lot of axons as white matter, right? So here we are again, kind of dissecting the nervous system into these binaries, right? Here's the gray matter, there's the white matter. But it does help us kind of organize our thoughts. And we can see that cell bodies and axons are in different places. So notice what they show here. Gray matter in the CNS is basically found in two places. I mentioned earlier the cortex, right? The very outer covering of the brain is going to be gray matter. Those are cell bodies. That's where the processing and the integration is taking place. We also have kind of clusters of cell bodies deeper in the brain, and we call them nuclei. I thought we already used that word as a part of a cell, but here we go. We are going to call clusters of gray matter deep within the brain nuclei. In the peripheral nervous system, we also get clusters of cells, and we call them ganglia. The plural there is ganglion, and so you will see that in a few of our upcoming chapters, right? In the central nervous system, though, we also have a lot of white matter, right? We see the white matter kind of throughout. Those are basically bundles of axons, right? All that myelin, all that insulation, this is axons, and we call those tracts. In the peripheral nervous system, those very same bundles of axons, sometimes they're the same axons. You will see where they cross from inside the central nervous system to outside, and it goes from being called a tract to a nerve, right? And you're probably used to that terminology of nerves. That's not confusing at all, is it? But try to get some of this terminology down, because you will definitely see nuclei, tracts, ganglia, as we move forward in our understanding of the nervous system. Okay, there are different types of neurons. This chart is showing you the structural classification of neurons, right? We can see that their structures differ, but they also differ in physiology. And so I'm going to mention some of those differences as we run through here as well, kind of what the functions of those neurons are. So let's start with this anaxonic neuron. Basically, this has lots of different processes, and you can't really distinguish who's an axon and who's a dendrite. These neurons functionally are very often what's called an interneuron. Right? Lots of them in the central nervous system's gray matter, right? Notice again, if you don't have an axon, you don't have that myelin, that white matter. These interneurons are super important in the processing and the integrating of information. Bipolar neurons are fairly rare, so we won't spend a whole lot of time here. We will see them in some of our special senses, particularly in sight. And what we're looking at, right, is we have a cell body right in the middle, a dendrite on one end, an axon on the other. This is an unusual neuron, but we will see it later in the semester. Unipolar neurons functionally tend to be sensory neurons. Okay. Here we see anatomically, right, the structure is that the cell body is off to one side. They call it a unipolar neuron because you basically have this one long axon and the cell body is shoved off to the side. So the dendrites are going to receive that information. We're actually going to start an action potential up here, and it can just keep going. Again, this is what we tend to find in most of our sensory neurons, those afferent neurons that are carrying information up to the brain. Multipolar neurons, this is what the prototypical neuron, the pictures that we've been looking at, are based off of. So here's our multipolar neuron, right? Cell body kind of up one end with all the dendrites and an axon reaching away. These are going to be our motor neurons. They take whatever decision all the anaxonic neurons made, and they're going to send it out to those effectors. So the cell body is up in the central nervous system, right? And it's going to send that message. It's going to exit that message through the efferent nervous system. Okay. Notice even in this picture that came a little bit earlier in the presentation that they're showing you this, right? We have sensory neurons going to the CNS and then motor neurons going away. Look at what their artist did. They drew this as a unipolar neuron for the sensory neuron. They drew this as a multipolar neuron for the efferent neuron. I don't love this inner neuron, right? It doesn't look quite as anaxonic as the ones we talked about, but that's fine, right? But see what we're trying to get at there. So afferent sensory neurons are unipolar, efferent motor neurons are multipolar. Okay. So that's kind of our big overview, right? Afferent division, integration, efferent division. And pretty much, we're getting there. One of the last things we need to look at are the neuroglia. Now, these are often just described as support cells. They have really important roles here in the nervous system and probably aren't that well understood. I think scientists have spent a lot of time trying to understand the neurons and how they're connected and how they make sense of things. But I've seen some research that indicates that some of these support cells might actually have some bigger roles that we just haven't figured out yet. So that could be kind of fun through your career. There's a book I read, I think it was called The Other Brain, and talking about how some of these neuroglia might actually be making decisions. Anyway, I digress. So what we need to know here is we have 4 different neuroglia, or some glial cells, same thing, neuroglia or glial cells. 4 different types in the central nervous system, 2 in the peripheral nervous system. In the central nervous system, the 4 types are astrocytes. These are named astrocytes because they're kind of star-shaped. And they can use all these little projections to hold on to a bunch of different neurons, kind of stabilize them in place. They also wrap around capillaries and are important in the formation of the blood-brain barrier. The blood-brain barrier is like this extra coating on the capillaries in the brain that makes sure that pathogens cannot cross into the brain and cause an infection. We do this extra layer of protection for the nervous system. They're also then, right, as part of that blood-brain barrier, going to do a lot of regulating what moves in and out of the blood. So what should the ion levels be? What should the oxygen or CO2 levels be, right? How much glucose is coming in, that sort of thing, okay? They also can be a total pain in the butt when it comes to fixing injuries in the brain. A lot of times they end up just producing scar tissue in their attempt to help repair damaged neurons. Our second set of cells are called ependymal cells. These cells are going to make cerebral spinal fluid. This is the fluid that kind of bathes the brain and spinal cord. Again, they'll play an important role in making sure that that fluid has the right ion and nutrient concentrations. They actually have cilia. They're helping to propel that cerebral spinal fluid throughout the spaces surrounding the brain and spinal cord. Here's a humdinger of a word: oligodendrocytes. Oligodendrocytes are what myelinate the axons in the CNS, right? So here we see, this is a difficult picture. This is the oligodendrocyte, and it reaches out these arms that then can wrap layer after layer of phospholipid, right, of fat around the axons of neighboring neurons. So they can cover more than one neuron at a time. And then finally in the CNS we have microglia. These are phagocytic cells. So they're related to macrophages. They are actually going to stay inside of the brain throughout their life. They're helping us recycle debris, trying to, if a pathogen happens to get through that blood-brain barrier, go ahead and engulf it and get rid of it. Before we go there, let's take a look. So your book has a couple really fun pictures, but they're kind of chaotic to just think about the cell types, right? So they're telling you kind of the same thing here of what the cells do. Let's look at this in the picture. So our astrocyte here, right? Notice all the little arms. Here it's covering a capillary, right? That's its role as the blood-brain barrier. But notice some of the other feet are reaching out and holding on to different axons and stabilizing them. The oligodendrocyte, we see wrapping. They've kind of got a little cutaway here so that you can see how many different layers of cell membrane of insulation you have. And that myelin, that insulation is important. Just like electric lines are wrapped in an insulation, it's going to help the action potential spread more quickly. Oops, sorry. Here's our microglia, our little phagocytic cell. And then ependymal cells are the ones, here they're showing you, this is called the central canal. We'll look at, there's some ventricles, some spaces in the brain. Those are all going to be lined in these ependymal cells that are making cerebral spinal fluid. Okay. This is a little bit of a detail showing you how myelination actually occurs, right? So our in this case, they're showing a Schwann cell. In this case, they're showing an oligodendrocyte. The Schwann cells are in that peripheral nervous system. Let me go back. Okay, so the two neuroglia in the peripheral nervous system, one group are called satellite cells. They're really similar, I would say, to astrocytes, right? So they're, again, regulating nutrients, gases, that sort of thing, kind of stabilizing cells. The Schwann cells are very similar to the oligodendrocytes in that they are going to produce that myelination. Of course, this time that myelination is happening in the peripheral nervous system, right, surrounding those axons. So notice one thing here, the Schwann cells, the cell typically wraps itself around a single axon. In myelinated axons. Oligodendrocytes are always holding on to a few, but the myelination, the multiple layers, ends up looking really similar. In the peripheral nervous system, we can also have Schwann cells that grab a bunch of different axons, right? And we would call these unmyelinated axons. Notice this is a myelinated axon, just so much insulation around it. These Schwann cells are really dedicated to insulating. Over here, this Schwann cell has like 10 different axons. It's kind of covering them, but it's not going to help the cells conduct as quickly as myelinated axons, right? So Schwann cells are involved in both myelinated and unmyelinated. It's just a slightly different amount of myelin coating there. Okay, now we're to the last thing. Repair in the nervous system. Now, when we were in the tissues, we talked about the fact that nervous tissue is famous, I would even say, famous for its inability to repair, right, to undergo that regeneration, and instead tends to undergo fibrosis. Absolutely, the central nervous system, it's very difficult. Part of that is the role of astrocytes. They're trying to help, but they often create scar tissue that gets in the way. The peripheral nervous system is a little bit better at repairing itself, particularly when you have a damaged axon. If the cell body is still fine, right? If the cell body is fine, but there's an injury out on the axon, what ends up happening is you get macrophages and things kind of coming in to clear things out. But if that Schwann cell remains, it leaves kind of a tunnel behind that axon can usually kind of follow and regrow and attach to the same cell that it was supposed to be contacting. I pulled a picture from another book that maybe shows that better. So here's the site of injury, the macrophages are coming in, trying to clear it up, and we see that empty tunnel, right, where the macrophages are clearing out the debris of that dead portion of the axon. But the cell body, as long as it has enough nutrients, right, it can create new membrane, new proteins, right? We know it has a lot of ER and ribosomes, and so that's what it does. And so here we see the axon working on regrowing. I think this is kind of fun. It's like sticking out all of these attempts to find the right way, right? But one of them finds the way down the tube left by the Schwann cells and is going to be able to make contact most of the time. And then we'll see that those other growth processes kind of atrophy and go away. You may have experienced this before. Have you ever had like a neuropathy where something is numb? Maybe those ski boots that were a little too tight and your big toe went numb. And I'm not talking about like that day. I'm talking like it was numb for a couple months and you're going, this might be a problem. What have I done? But sure enough, at some point in the summer, you're like, wait a second, I can feel my toe again. This is exactly what happened. You had damage, right? But the neuron was, the axon was able to regrow. So we made it sound really bad in the tissue because we were saying basically these neurons don't undergo mitosis. If a cell dies, it's going to be dead. We're not getting it back. But a lot of damage that happens to us, particularly in the peripheral nervous system, is really in the axons, and they are capable of repairing as long as that damage isn't too extensive.